So dear students, today I am just going to start the new topic, that is the pollen grain or the microspore. Remember, I am just going to say the pollen grain or microspore. I am not going to say pollen grains or microgametes. So these are the two different terms. Pollen grains are the microspore and male gametes. Male gamete is quite different from the pollen grain or the microspore. So what is the difference between them? We'll see the later. So just start with that. Till now we have understood the process of formation of the pollen grain, like that in the process of microsporogenesis and the process of macrosporogenesis. So listen, what I'm just going to explain you. So the pollen grains are microspore. Till microsporogenesis we have just completed the formation of microspores. Now I am just going to make the detail of that structure. Okay, what is the microspore or pollen grain, how it is being recognized, what is the structure of microspore. So let's see. The shape of microspore or the pollen grain is globose or oval or spherical. Globose, the globular shape; oval, oval shape; egg shape, egg-like; and spherical. Spherical also means what? Somewhere it may be the polyhedral also. So the four different type of the shapes, either globose or oval or spherical or polyhedral. The size of the microspore or pollen grain is 25 to 30 micron. The single nucleated structure, the uninucleated structure it is. And one more very important question for the competitive examination. The pollen grain study is now being very vast in the botany, in the field of botany. So that is why a separate branch of the botany has been established, where we are dealing with the study of pollen grains only. So the study of pollen grain is called as palynology. Is it clear to you all? The study of pollen grain is called as palynology. Now we are just going to see the structure of pollen grain. The structure of pollen grain, the pollen grain or microspore is usually a kind of cell, a complete cell, where the cytoplasm is there, where nucleus is there, where the covering is there, and one important structure is there. So in a cell, in a microspore, the cytoplasm is present, the nucleus is present, the envelope is present. The envelope of microspore is very important or very crucial structure, that is sporoderm and the germ pore. So we'll see one by one. Nucleus, already I have just discussed, this is a single nucleated structure, only one nucleus is present. You can see there in the diagram also, the one nucleus is there. Along with that, a dense cytoplasm is present there in the pollen grains. Next one is sporoderm. So we are just taking the sporoderm in very detail. The sporoderm is sporo, microspores, derm. If we are just going to understand the etymology of that, so sporoderm means what? The walls of pollen grain. Sporoderm means what? Wall of pollen grains. So whatever the external covering, whatever the envelope present around the pollen grain is called as sporoderm. So is it clear to you all? The pollen grain is covered externally by a thick layer or covered externally by an envelope. That envelope is called as sporoderm. So that sporoderm is made of two layered, that sporoderm is a two layered structure, or we can say it is made of two layers, the outer layer and the internal layer. The outer layer of sporoderm is called as axine. Outer layer of sporoderm is called as axine. Listen, outer layer, the axine is the outer layer. It is very thick walled, rigid and ornamented. All points are very much clear to you all. Outer layer, axine is the outer layer, the thick walled structure, thicker than the internal one or inner one. Rigid means tough structure. Ornamented means variously modified and give a specific pattern or specific shape or specific structure to the pollen grain. This makes the basis of taxonomy also. Okay, what kind of the ornamented? Ornamented means fashioned. Ornamented means a type of shape or we can say ornamented means what kind of the pattern of the axine is present. So that is a basis of taxonomy also. So is it clear to you all? Outer layer, the thick walled, the rigid one, and ornamented, variously designed.
Start.
So now the word ornamented is clear. Ornamented, variously arranged axine gives the specific shapes and appearance to the variously specific shapes and appearance to the pollen grain. So now I'm just going to discuss very important point. I'm just going to discuss very important point that the exine is made of very important molecule that is called as sporopollenin. My dear child, my dear student, this is very important question, whether it is the NEET examination or it is the CBSE class 12th examination. Remember, the exine is made of an sporopollenin, made of a material sporopollenin. Many of the time the question is being asked there in the CBSE as well as the NEET examination. What is sporopollenin? Sporopollenin is a highly resistant organic polymer. Organic polysaccharide is there that is highly resistant. So very specific property of sporopollenin is that this is non-biodegradable, that cannot be degraded by the biological agencies, not by the any of the enzymes, not by any of the chemicals whatever is present in the environment. It is highly resistant for the high temperature. Strong acids present in the atmosphere, strong bases present in the atmosphere means it cannot be easily decomposed. It remains as it is, whatever or however it is present therein. And even though no enzyme so far has been reported till date that can degrade it. Are you getting my point? The sporopollenin is a tough organic polymer which is non-biodegradable, resistant for the high temperature, the strong acid, strong bases, and no enzyme so far is reported till date which can decompose the decompose the sporopollenin. So I think you can understand well कि if these enzymes are there, if these are the factors like that, any of the thing, any of the materials can decompose by the temperature, can decompose by the strong acidic reaction, can decompose by the strong basic reaction, and by the biological agencies. All 3-4 agencies, the sporopollenin remains unaffected from all these 3-4 agencies. So sporopollenin is a kind of tough material which cannot be degraded easily, and this property of sporopollenin makes the pollen grain suitable as a fossils. This is a shocking fact, but we have received the fossils of the pollen grains also. So suppose if any question is there in your examination कि why or which property of the pollen grain makes the suitable to pollen grain to work as a fossil, or why the pollen grains from the previous time or ancient time has been recovered successfully and we are able to study them properly, just because of presence of sporopollenin in them. Sporopollenin make them undegradable, non-degradable. So listen, always careful. And one more thing, if any more aspect is being demanded कि why the fossil nature of the pollen grain is required, so that is required for the future study. If we have to preserve the pollen grains of future study, we can forecast the fossil fuel also with the help of that. So the first layer is clear to you all. The sporoderm is the wall of pollen grain that is two-layered. The outer layer is called as exine, which is thick-walled, ornamented, rigid, thick-walled, highly thick structure, rigid, the tough structure, ornamented, variously modified, and this ornamentation makes the basis of taxonomy also. That exine is consist of a very strong polymer, that is sporopollenin, which is non-biodegradable, resistant against the high temperature, strong acid and the bases, and no enzyme can decompose it. That is why the pollen grains are available in form of the fossils also. So this is very much clear to you all. Now I am just going to put some more light, or I am just going to make some more light on the exine. So okay, so next to that, the exine is itself a two-layered structure. If you are just going to make some detailed study of the exine, so exine is made of two layers. The outer layer is called as sexine, and the inner layer of exine is called as nexine. Means what? Suppose this is the exine. So exine is itself made of two layers. The outer one is sexine and inner one is called as nexine. The sexine is itself made of two layers, endosexine and tectum. Endosuction is the inner layer and tectum is the outer layer. Are you getting my point? First of all, exine, first of all, pollen grain is made of two layer, that is exine and intine, but exine is itself made of the two layer. The sexine is the outer one. The sexine is the outer one and sexine is itself made of the two layer, that is endosuction and tectum. The outer one is called as tectum and the inner one is called as endosuction. The inner layer of the exine is called as nexine. The nexine is once again made of two layer, that is endaxin and foot layer. Endaxin is the inner one and outer one is the foot layer. So if I am just going to see the hierarchy of these layer, so the number one layer which has just come from the outside is T, means tectum. Tectum will come with the endosuction, then endosuction will be followed by the foot layer. The foot layer will be followed by the endaxin, and finally that will followed in form of intine. So this is the hierarchy. Remember, this hierarchy is crucial for the higher order questions there in the NEET also. Ki what are the layers? The outermost layer of the pollen wall or sporoderm is the tectum. The next one is endosuction. The third layer is the foot layer, then endaxin, and finally the intine. Now the next layer of the pollen grain is entine. The entine is the inner layer. As comparison to the exine, this is soft and elastic. Just vice versa to the nature of exine. Exine was the tough, resistant, hard, thick walled, but here entine is the inner one, soft and elastic. The important question which can be asked from here, that is the combination of intine. Intine is a pectocellulose. Intine is composed of a pectocellulose material, as exine was composed of the sporopollenin. Similarly, it was the intine is composed of the pectocellulose. What is pectocellulose? Pectin and cellulose. These two materials combines to form the pectocellulose, and intine is very much important. Why this is important? Remember, this is also once again very important point. What is the role of intine? Exine is going to prevent or protect the pollen grain. Exine is going to protect the pollen grain from the external harsh environment. Due to that, the pollen grain remain viable for so long time. But what is the role of intine? The role of intine is the intine is responsible for the development of pollen tube. Are you getting my point? The intine is responsible for the development of pollen tube, and in that pollen tube only the formation of male gamete occur, that we'll discuss in the next lecture. So is it clear to you all? Remember, exine is protective in the nature and intine is important for the development of pollen tube. Is it clear to you all? Just next in the structure, as we have already discussed, what are the structure of the pollen grain? Number one, the cytoplasm. Number two, the nucleus. We have completed cytoplasm, we have completed nucleus, we have completed the sporoderm. Sporoderm is important. The outer one layer is called as exine. Sporopollenin works for the protection from the strong acid, bases, temperature, resistant against so many harsh things, and intine is elastic, made of pectocellulose and continues in form of the pollen tube. Now next one is the germ pore. What is the germ pore? Just listen carefully, just try to understand. The exine is a tough layer. If exine is present on all around the microspore or pollen grain, so this makes very difficult the process of germination, process of microsporogenesis, because the process of germination and microsporogenesis demands the growth of pollen tube. So to explain or to ensure the growth of pollen tube, to ensure the germination of the pollen grain, this is required that exine should not be present on everywhere. This is what the nature has created an arrangement. The some of the places on microspore remains very thin exine are almost absent exine. We can say the places where exine is absent are very much thin. These places are called as germ pore, because from here the pollen tube will just come out. Are you getting my point? Some of the places on the microspore or pollen grain, the exine is very thin or just remains absent. Those places are called as jump over. This is a kind of very big discussion, big debate, that whether the exine is absent or whether the exine is very thin. Many of the resources are writing that exine is absent, many of the resources are writing that it is thin, but we are just taking both the views, both the references here. But in my view only, the absent word is not proper. If the exine remain absent, then what about the viability of the pollen grain? Due to the exine only they viable for the longer time. If exine remains absent, they may just decompose earlier only. So what is the importance of germ pore? Once again I'm just coming back. Germ pore is the place from where pollen tube comes out from the pollen grain. Pollen tube comes out from the pollen grain. Tomorrow only in the next lecture we'll see how the pollen grain comes out. So is the point is clear to you all? I think the places where exine is almost absent or it is very much thin, that is called as germ pore. And what is the importance of germ pore? The pollen tube from the place from this place only the pollen tube comes out. So how many germ pores are present in a pollen grain? That is also very important question in the NEET examination. Remember, normally the first time whenever the germ pore develops, the germ pore develops in form of a simple furrows. Those furrows, since they are just going to gravize the germ pore, so they are called as germinal furrows. Are you getting my point? Due to the initial time or during the initial time of developing the pollen tube or germ pore, the germ pore originate earlier in form of the germinal furrows. In dicots, we are just taking again the example of capsula, because yesterday only in the previous lecture I just explained you the capsula is the most studied pattern of the embryonic development in dicot. So most of the dicots follows the same pattern of the capsula. So in dicots they have three germinal furrows. The dicots have three germinal furrows. Means what? These all three germinal furrows will get converted in form of germ pores. What I am saying, the dicots capsula have three germinal furrows, which in turn just getting converted in form of germ pore. So how many germ pores are present there in the dicot? Three germ pore. So if the pollen grain is having three germ pore present there, so that type of pollen grain is called as tricolpate pollen grain. Is it clear to you all? If the dicots having three germinal furrows, these germinal furrows will get converted in form of germ pores. So dicots will have three germ pores, and such kind of pollen grains are called as tricolpate. Just opposite to that, the monocots have only one germinal furrows. Some of the references, some of the resources are writing that there is no germ pore present in monocot, only they are having the germinal furrow. But if the germinal furrow is there, then it will be considered as the germ pore also. So monocots have only one germinal furrow, means only one germ pore. That's why that is monocolpate. Tricolpate in dicots and monocolpate in monocots. So is it clear to you all? I think now this is the time to understand the diagram structure with the help of diagram. So I'm just going to explain with the help of diagram. Okay, so whatever we have explained till now, I'm just going to show you with the help of very simple diagram. The nucleus present here. The nucleus is surrounded with the thick and dense cytoplasm. Outside these two layers are going to form the sporoderm, means exine, and intine will form the sporoderm. The sporoderm, exine is the outer one layer. You can see that is variously ornamented. A design is different, arrangement is different. The inside structure is called as intine. Some places the exine is very thin, or we can say it is absent. Those places are called as germ pore. This is very smart, very simple structure of the germ pore, but your final examination demands this structure. So please be ready with that structure. The structure of pollen grain. What is that? This is the structure of pollen grain, not the male gamete. This is the structure of pollen grain or microspore. What is very simple? Nucleus, cytoplasm, intine, exine and germ pore. All four points are very much clear to you all. So now we are proceeding forward to this. So after completion of the microspores and their structure, now the microspores are ready to liberate out. How they are just coming out from the microsporangium or the pollen sac. The process of coming out of the pollen grain from the microsporangium is depending upon bursting of the anther. So bursting of anther is called as dehiscence of anther. Are you getting my point? Until and unless the microsporangium or the pollen sacs are getting intact, the pollen grain will remain inside only. But this is required that they must release out from the pollen sac or embryo microsporangium. So bursting out, blasting out, damaging, destruction of that anther lobe after maturation of the pollen grains, after formation of the pollen grain, is called as dehiscence of anther. How the dehiscence occurs, just try to understand. I'm just going to explain it in very simple way, in very easy way, by the help of a flowchart. Just pay your attention here. As soon as the maturation of pollen grain occur, means the pollen grain is now having well defined structure, nucleus, sporoderm, everything is present there. As soon as the pollen grain matures, they are just going to completely dries up. The pollen grain, when they are just getting matured, probably they are just going to dries up, and they are just converted in form of a pollen dust or the pollen powder. Are you getting my point? After maturation of the pollen grain, they are just getting dried up and finally getting converted in form of the pollen dust or the pollen powder. Is it clear to you all? This is the first step. After maturation, they are dries up and converted in form of the pollen dust and the pollen powder. Next to that, what happens just after maturation of the pollen grains? Now one by one, all the layers, after maturation of the pollen grains and conversion of them in form of the pollen dust, one by one, all the layers of the parietal wall layer, means what? Tapetum, endothecium, wall layer, everything are now going to be degenerate. First of all, the middle wall layer, since they are the parenchymatous cells, they are being absorbed in form of the nutrient with the help of tapetum. So since they are being completely absorbed during the maturation of the pollen grain, so they are just getting degenerate. The first of all, which layer just getting degenerate? Middle layer. The first of all, which layer just getting degenerate? The middle layer. Secondly, which layer is going to degenerate? The tapetum. Remember, the first of all, which layer is going to degenerate? That is the middle layer. The second one is the tapetum. Now the dehiscence of anther or release of pollen grain, depending upon the dry season, they are just going to dehiscate out in the dry season only. Dry season means where very less moisture content is available there. So what happens in the dry season? The endothecium, only two layers now remains in the mature pollen grain. Remember, only two layers are there. Number one, two layers. Number one is epidermis and number two is endothecium. Only two layers are there. Rest of the layers are getting degenerated. Just now I have just explained, tapetum and middle layer, they are consumed due to provision of the nutrition. So as soon as the dry season comes, now the endothecium also just getting degenerated due to the loss of water. Dry season is there, so moisture content will be evaporated from the endothecium. Due to the evaporation of the moisture content from the endothecium, they are also getting shrinked. Due to the shrinkage of them, the cells of the endothecium are getting died. The cells are getting died and they are just going to create a pressure on the stomium, and the stomium will get burst out and the pollen get released out. Are you getting my point? Once again I'm just releasing, once again I'm just explaining. Due to the dry atmosphere, the moisture content of the endothecium will just evaporate out. The moisture content evaporate out, the cells of endothecium get shrinkage, they are just getting died, they are getting rupture, and due to the rupture of them, the pressure will be exerted on the endothecium. The pressure will be exerted on the endothecium, and due to that pressure the stomium will result in the bursting, and due to the bursting of the stomium the pollen grains will release out in the atmosphere. That is what the dehiscence of anther. Once again I'm just going to explain the complete process of the dehiscence of anther. The dehiscence of anther is what? First of all, the maturation of pollen grain occur. After maturation, they are just drying and getting converted in form of pollen dust or the pollen powder. During the maturation, what happens? Two layers, the tapetum and the middle wall layer, completely digested, completely degenerated due to the provision of the nutrients when the dry season comes. But if you can, if any question here is there, ki how many layers are present there in the mature pollen grain? So remember, only two layers are present in the mature pollen grain. One is epidermis and one is endothecium. As soon as the dry season comes, what happens? The evaporation of the water occurs from the endothecium also. Due to that, the endothecium gets shrink, died, and will create an extra pressure on the stomium that will get burst and pollen grains will release out. That is what the dehiscence of anther. Now the pollen grains are liberated in the atmosphere. Now they are ready for the pollination purpose. Remember one very important point I'm just going to explain. I'm just going to say, you must note down it carefully. The angiosperms have the longitudinal axis dehiscence, means the dehiscence in the angiosperm plants is longitudinal, means the capsula is having the longitudinal dehiscence. Angiosperm, we can say the example of capsula once again. So capsula is also having the longitudinal dehiscence. So I think today it is very much clear to you all what is the structure of pollen grain, how the pollen grains are now liberated in the atmosphere. In the coming lectures, we'll understand how these pollen grains are reaching onto the surface of stigma and how these pollen grains are now just going to develop the microgametes. So just wait for the next lecture where we'll understand the development of male gametophyte along with the development of microgametes. Thank you.